Hello, today I'm going to be going over the Unit 5 study guide with you for your biology exam. Unit 5 is about the cell cycle, and in the cell cycle we talk about cell division and how cells make new cells. Remember from Unit 1, we said that cells only come from other existing cells, and they're the basic unit and of structure and function in living organisms. So it's important to know how they're made and where they come from. The two main ideas in this unit were the ideas of mitosis and meiosis. So big things to think about when you're studying are what are the differences and similarities between mitosis and meiosis. Taking a look at the study guide. Number 1 says the sex cells that are produced during meiosis are what? Remember, the E in meiosis is for sex. These cells are unique, meaning that they're not identical, they're different from each other, and you get a half set of cells, of chromosomes in those cells. Meiosis is only producing sperm or eggs. It's only producing sex cells. Number 2, in what cellular division process is nuclear material divided? So in other words, the contents of the nucleus is divided during mitosis. What occurs during the interphase stage? That's going to be your G1, S, and G2, and that is when cell growth, DNA replication, and checking, the enzymes checking to make sure everything is correct. That's what's going to happen during interphase. So that's primarily cell growth. Number 4, how are mitosis and meiosis similar to each other? They both are a process of cellular division, so they both make new cells. Number 5, what does the interphase, prophase, metaphase, anaphase, telophase, and cytokinesis stages of mitosis look like in a cell? Over here where we have interphase, you see you have your cell membrane, the nucleus, and the cytoplasm. During prophase, your nuclear envelope is going to start to break down and your DNA is going to condense into chromatid. During the metaphase stage, in the M, we think of middle. The chromosomes line up along the middle or the equator of the cell. During the anaphase, remember Ana? You broke up. Anaphase is the breakup, the splitting up of the chromosomes. Telophase is when the cytoplasm is going to pinch in a little bit and two new nuclei are going to start to form. Cytokinesis is the division of the cytoplasm. This is where the cell formally splits and you now have two completely new nuclei. They should look exactly like the original cell. They're identical. Number 6, during mitosis, how many daughter cells are produced and are they identical or different? They're identical, and we're making 2. How many cell divisions does mitosis have? 1. How many cell divisions does meiosis have? 2. Which method, meiosis or mitosis, produces sex cells? That would be meiosis. What happens during cytokinesis? That's the division of the cytoplasm. Now for number 11, I'm going to come back to this one because I'm going to take the camera down and I'm going to show you a picture on the computer. It was a little bit hard to draw in that part of the paper, and I don't think it would come up on the screen. So I'm going to come back to number 11. Number 12, in meiosis, when the sex cell divides, how many chromosomes are in each daughter cell? Half. And the half stands for haploid. What happens to the genetic information in the cell before it divides? That genetic information is going to copy itself, remember, during the S phase. That stands for DNA synthesis. Daughter cells produced during mitosis are identical to each other. Daughter cells produced during meiosis are diverse, meaning they're different. What is mitosis's direct role? The reason a cell goes through mitosis, it's only your somatic body cells, so every cell except for your sex cells. It's going to go through mitosis for growth and repair. What type of cell undergoes meiosis? Just sex cells, sperm or eggs. The main purpose of mitosis is to divide the contents of the nucleus, so it's called nuclear division sometimes. What number of chromosomes are in a new cell formed through mitosis? It's going to be the same number. For humans, it'll go from 46 to 46. If you were a fruit fly, it would go from 8 to 8. During the interphase stage, you have G1, growth, S, which stands for DNA synthesis, to make new DNA, and G2. It's checking to make sure everything's how it should be before the cell enters mitosis. Number 21. When is DNA replicating during the cell cycle? This is a tricky one because if you think about it in terms of it being a cell cycle, it happens after mitosis occurs, after G1. You could also say that it happens before mitosis occurs, depending on which direction you're looking at it. So it's a little bit of a confusing question there. 22. During mitosis, the daughter cells their chromosome number is going to stay the same. 23, 2 cells are produced by the end of mitosis. 24, 4 genetically different haploid cells are produced at the end of meiosis. And they're going to be sperm if you're a boy or eggs if you're a girl. Those are their only 2 choices. What step of meiosis would be what steps in meiosis would be most likely to have chromosomes have a failure to separate? And that would be anaphase. Remember, metaphase is when they're all lined up together along the equator of the cell. Anaphase is the breakup. So if something was to go wrong and chromosomes failed to separate, they would have that mistake happen during anaphase. Number 26. How is chromosomes separation in meiosis different from chromosome separation in mitosis? And we've been saying this over and over again. It's half. You have half the amount of chromosomes coming out of meiosis. Number 27 is a little bit of a critical thinking question. It says, compare the effects of a disorder in which a chromosome fails to separate in meiosis to chromosomes failing to separate in mitosis. If you have an error occur in meiosis and you end up with, let's say, 24 chromosomes in an egg instead of 23, that genetic defect could be passed on to future generations because it's in one of your sex cells. So chromosomes failing to separate in meiosis happens to sex cells. That means it's something that could be passed on to future generations. On the other hand, if something goes wrong when, let's say, a skin cell is dividing or a muscle cell is dividing, that's not necessarily going to be passed on to future generations because it's occurring in your regular or somatic body cells. All right, bear with me for a second while I take the camera down, and I'm going to go back to number 11. Number 11 was this picture, wanting to see the difference between the processes of meiosis 1 and meiosis 2. So I'm going to take this out and try to film it. I want to show you the picture from the slideshow. All right. In meiosis 1, we call this the reduction division phase. It looks just like regular mitosis, but if we look closely, we see that in prophase of meiosis 1, the chromosomes do something very important. They cross over. That means they kind of just rub on each other and swap information. What that's going to do is it's going to scramble your genetic information just a little bit. Now when these chromosomes split, they won't be identical copies to each other the way that they are in mitosis. They're going to be a little bit different. So crossing over provides a little bit of genetic variation among the chromosomes of your cells. So that's going to help you to not be a clone of your brothers and sisters. So meiosis 1 is called the reduction division. There's your chromosomes, prophase 1. Later in prophase 1, we have the crossing over occur. Metaphase 1 is the same, middle. Anaphase 1 is the breakup. And now we have 2 cells that have fewer chromosomes than the original. See how there's 4 in there? And each of these have 2. So we've reduced our number. We've also scrambled our information a little bit. So that's meiosis 1. The real important part there to think about is the crossing over that occurs in prophase of meiosis 1. In meiosis 2, those 2 cells are now going to go through the process again. Prophase 2, metaphase 2. There's no more crossing over here. Anaphase 2, telophase 2, and then finally we end up with 4 different haploid daughter cells. Okay? Before we go, I wanted to let you know, short answer questions on your test are going to include a picture of the cell cycle, and I'm going to want you to be able to name and label the parts. So you're going to need to know G1, S, and G2. And then I would like you to enter the stages of mitosis in the diagram as well. Prophase, metaphase, anaphase, telophase, and then cytokinesis. All right, I think that's it. Good luck on your test. I hope this was helpful. And if you have any questions, don't forget to ask me.